Okay, welcome everybody. My name's Jerry Pritchard. I'm a professor at University of Birmingham and also a member of RSB, and I'm chairing this RSB Plant Health Series event today. This is in partnership with DEFRA, and we aim to continue to develop the diverse community from across the sector to build UK plant health capability, resilience, capacity, etc. Really important thing these days with all the biosecurity issues. So we've got two linked speakers today, and as we go, I'll introduce those in a second, but as we go through these two linked talks, please, if you know how this works, put your general questions in the Q&A box and we'll sift through those and answer those at the end. There'll be time at the end to put those questions through, but continue to do that all the way through. And if you fancy it, please post to any of your social media channels with hashtag RSB Plant Health, which would be very welcome. Okay, so without further ado, I'll introduce the speakers. The first speaker is Lorena Rangel, a plant pathologist who works on the fascinating world of plant microbiomes. We all know about our own microbiome, but did we know about plants? It's for bacteria and fungi living on crop leaves and their wild relatives. Her research explores how these tiny communities change during disease and how we might use them to develop smarter, more sustainable ways to protect plants. Lorena will share her journey into academia, so a careers-based thing as well, and the international research experiences that shaped her career. She'll give us an inside look at her work on bacterial ecology, plant microbiomes, and how these interact with fungal diseases. She'll also talk about her role in the Integrated Pest Management group at the James Hutton Institute, where she and colleagues are searching for alternatives to traditional fungicides. And of particular interest to this group, she'll update us on the FUGS RSB project she led in the summer of 2024, what's happened since, and where her research is heading next. I'm also very pleased to introduce Alicia Showering, a molecular microbiologist whose research has led to exciting breakthroughs in natural pest protection, presumably national as well. During her PhD at the London School of Hygiene and Tropical Medicine, Alicia discovered that some microbes can naturally repel pests without disrupting beneficial insects or the wider environment. That finding ultimately sparked the creation of her company, BugBiome. So in her talk today, Alicia will share the story behind funding BugBiome, from an early internship to shifting her focus from human microbiomes and mosquito research to innovative crop protection solutions. She'll also explain why cultivating a diverse range of microbes is so important and how her team sources them and why traditional insecticides are losing effectiveness, highlighting the need for sustainable nature-based alternatives. All very topical, all very exciting, and without further ado, I'll pass over to Lorena. Lorena, please.
Thanks for that introduction, Jeremy. Let's see. How does this look for all of you?
That looks fine. Yep.
All right, great. Let's see if I can move this. All right, thanks for that introduction. So my name is Lorena. I'm going to talk to you today about phylosphere ecology and sustainable crop protection. So the work that focuses on understanding leaf-associated microbial communities and how we can translate that knowledge into sustainable agricultural applications, particularly within UK systems. So I'm just going to give a little background on myself. I grew up in the US between northern Minnesota and southern Texas. I completed my undergraduate degree in microbiology at Texas State University. Then I moved to Oregon, where I completed my plant pathology master's degree at Oregon State, and moved afterwards to do my PhD at the University of California in Davis. And then it was kind of a full circle for me. I headed to a postdoc within 5 miles of where I was born in Fargo, North Dakota, with the United States Department of Agriculture. And that was just before coming to the UK, where I've joined at the James Hutton Institute in Scotland in the Cell and Molecular Sciences Department. So my background really spans plant pathology and microbial ecology, and that'll directly inform the work that I'll show you today. So I've always had a focus on the leaf surface as a habitat for microbes. we call this the phylosphere, and actually that refers to the all of the above ground plant surfaces as a microbial habitat. So this is a highly exposed environment. There are many stresses like temperature fluctuations, for example, but despite that, it hosts a diverse microbial community. So these pressures are really selecting for organisms with specialized traits. This includes stress tolerance mechanisms, specialized enzyme production, and diverse secondary metabolites. So the phylosphere is a really promising and relatively underexplored source of bioactive metabolites that can be used in many industries, including agriculture. So how do we translate this potential into real crop systems? In the UK specifically, well, globally actually, crop systems are under pressure to reduce pesticide reliance. So that shift from ecological discovery, for example, on the phylosphere, to agriculture application becomes especially urgent. So before discussing pesticides pressures, I think it's important to understand the role of UK cropping systems. So the UK has approximately 25 million hectares of total land, and about 6 million hectares are devoted to arable crops. So that means roughly a quarter of the country's land is used for crop production. And the majority of cropland is planted with cereals. So this will be your wheat and barley mostly. And then there's also oilseed, potatoes, vegetables, and soft fruits that represent important fractions of production. As of 2025, the statistic just came out last October, UK crops represent about a 12 billion pound industry. And so this scale really highlights why sustainable disease management is of national economic significance. So given the scale and economic importance of UK cropping systems, maintaining yield stability is really important. And these crops are constantly under increasing constraint. So we're seeing a loss of active ingredients due to regulatory withdrawal. There's growing pathogen resistance to existing chemistries. And as you can see here, we recently had a national action plan that discusses working for a more sustainable future and how we work with pesticides. And it discusses about reducing overall pesticide risk by at least half just in the next few years. So as a result, there's a growing need for alternative strategies that align with integrated pest management. And so that's actually, if you can highlight here in objective one, that's the first step they're looking for. And so IPM, or Integrated Pest Management, prioritizes prevention and low-risk methods before chemical intervention. And so it aims to optimize sustainability while minimizing environmental damage. So industry already has marketable microbial products. So many of these are bacterial-based. Many of them are through major companies such as Syngenta and Corteva. But really a critical issue with them is that there are more products that are registered than are effective or even used by growers. So this gap between registration and field efficacy and use shows the need for deeper knowledge in the ecological understanding of plant microbiomes. So before discussing pathogens directly, I think it's important to also consider the ecology of the leaf. So the leaf microbiome exists in, like I mentioned, extreme and ephemeral environments. So these microbes will experience daily fluctuations in temperature, UV radiation, water availability, and desiccation across a plant's entire life cycle. And the community is shaped by abiotic filtering, host-driven biotic factors, and interactions among the microbial residents themselves. And so the residents include bacteria, fungi, archaea, algae, and viruses. So this is really a multi-kingdom system on the leaf surface. And these interactions are really the baseline for understanding how pathogens can further reshape these communities. As a plant pathologist, interactions between the foliar pathogens and the resident microbial community are really how I think about disease. Disease is often associated with reduced microbiome diversity. So it's been shown that foliar pathogens actively reshape their niche through secreting effector proteins or secondary metabolites that will alter the community structure on the leaf surface. And these interactions inform how I think about beneficial microbes as part of biological control strategies. And I think a really important question is, are the microbiome shifts a consequence of disease, or can microbial communities influence disease outcome? So to explore this in practice, I'm just going to give an example of during my postdoc where I focused on Cercospora leaf spot and sugar beet. So this disease is caused by the fungus Cercospora beticula. It's the most damaging foliar disease of sugar beet globally. And this pathogen is well known for its range of secondary metabolites that contribute to host tissue damage. And if left uncontrolled, sucrose yield losses can reach up to 50%. So it's really devastating. It has major economic consequences. And currently, management relies on fungicide applications, crop rotation, and the deployment of resistant cultivars. However, fungicide resistance in Cercospora populations is an increasing problem. And so alternative management strategies have been necessary. So just to give some context about the crop, the majority of refined sugar in the US is derived from sugar beet, so about 60% of the sugar in the US. And the majority of this crop is grown in the Red River Valley, which is this circle shown on the map here. So this is the border of North Dakota and Minnesota. So using a field here where we had Cercospora leaf spot resistant and susceptible cultivars planted, we allowed the crop to naturally acquire the disease. And then we looked at the bacterial community on the leaves over the growing season and asked the question, does the foliar bacterial community shift with disease development? And can we use these native microbes to combat disease? So rather than introducing a foreign biocontrol agent, we focused on the native phylosphere residents that were already adapted to this environment. And so we analyzed bacterial communities from the sugar beet leaves across disease progression, so early, mid, and late season. And so these are just representative images of leaves and the plots. And you can see by just pre-harvest, the susceptible plot and leaves are completely necrotic. If you look at panel C, this is a PCA plot. So this shows samples that are clustered by similarity. So think of it like the microbial community makeup and how that changes over time. And what we observed was that the communities really diverged significantly once visible symptoms developed. And so these disease leaves showed altered composition compared to the asymptomatic leaves. Importantly, however, so the resistant and susceptible cultivars still shared a core microbiome. And so this means that there was a stable group of taxa that was consistently associated with sugar beet, regardless of genotype or disease. So in this red box are the genera that were found at greater than 1% abundance. So this just shows the major players that are present. And this is the core community that was always present, just at different ratios among the varieties and times. And so to move beyond just sequencing and descriptive profiling of the community, we also in tandem created a sugar beet associated bacterial culture collection. And we, or rather the master's student at the time, Madison Christensen, who just graduated, isolated strains representing 28 unique genera. And the dominant genera included your pseudomonads and syngomonads. But these are the taxa that were commonly found in the core microbiome in the previous slide that I just mentioned. And the goal with these was to translate sequencing observations into functional assays. So by culturing these representative strains, we could directly test them on whether they had an effect on Cercospora growth. And so in the lab, she performed antagonism assays against the fungus. And several of the isolates inhibited fungal growth compared to the controls, and the inhibition varied by strain. But this indicated that there are native sugar beet microbiome bacteria with measurable antagonistic activity. These results supported the hypothesis that disease-associated shifts aren't necessarily a passive consequence of infection, but might reflect competitive interactions that are happening directly on the leaf surface. And I think the broader implication to think about is that the native phylosphere communities do contain strains with biocontrol potential that are already environmentally adapted to that specific crop. And so I've brought that approach to the James Hutton Institute. We're located in Dundee, Scotland. And the Institute has facilities spanning genomics, imaging, metabolomics, and translational research. So we have our glass houses and fields right here on campus, which is great because it's access to all kinds of things. So if anybody's interested in collaborating, please contact me. But what we're doing now is applying that kind of a crop agnostic approach. So we're profiling native microbial communities, isolating strains, and trying to link mechanisms towards that antagonism and translate these findings into integrated pest management strategies. And so one focus system that I'm working on is barley and rhynchosporium, which is the causal agent of barley scald. And so we run a field-based microbial discovery at our rhynchosporium nursery. So this is a site on campus that's been used for barley breeders for about 40 years to determine disease ratings for barley scald. And because we can't inoculate the fungus directly into the field, this site is really great because it has a buildup of inoculum, right? So we can plant barley that we know will be infected with scald and isolate microbes that are directly from the leaf surface and evaluate them as potential biocontrol agents. And so I had a summer student who helped us create this culture collection. And you can see on the left, the morphological diversity. This is just on a plate of bacterial colonies growing. And then on the right is a phylogenetic tree showing the genetic diversity among these isolates. So from this culture collection, we've started to identify strains that have biological control potential. And so this got us in collaboration with Alicia from BugBiome, who you'll hear from next. And we asked the question, can enrichment of leaf microbiota using extracts from pests or pathogens enable cultivation of previously unculturable microbes? So we use extracts from insects, plants, and fungi within media to see how the bacterial community changed compared to media alone. And I think it's important to say that a major bottleneck in microbiome research is that sequencing will show us diversity, but much of what we see in this diversity hasn't been cultured. So without cultivation, we can't validate function or develop products. And for industry, this is super important because diversity really only becomes valuable when it can be grown, tested, and translated into usable strains, which is what this enrichment approach aimed to achieve. So we secured funding from SALSA, where we were able to test enrichment using insect, fungal, and plant-derived extracts. So all this data that I'm about to show you is very new. We just started analyzing it, but what we measured, one measurement we had was bacterial diversity. So the graph you're seeing is a Shannon index. So this will capture both richness and evenness of these communities. And so think of it as the higher the value, the more diverse the sample. So as you can see in the second and third box plots, insect and fungal enrichments really increase diversity, which surprisingly, plant enrichment relatively reduced diversity compared to the media alone or media with antifungal selection. So this just showed that the substrates can directly shape community structure, and the increasing diversity suggests that we're expanding detectable and maybe culturable fractions of the microbiome. And so we also examined bacterial composition to determine how enrichment altered bacterial community structure. So the treatments didn't just change the diversity, but they shifted which taxa were dominant. And so if you look specifically at the insect and plant enrichments, these promoted a unique subset of microbes. And so this just showed us that enrichment can act as a selective pressure to reshape the community composition rather than just increase abundance. And then we also used metagenomics, which sequences all the DNA in a sample and examined which genes are collectively carried within a given sample. And so we found that all the genes amongst all the samples had approximately 115,000 genes shared amongst them. But if you look at the specifically enriched samples, there are about 13,000 to 22,000 unique genes after the enrichment. And so this shows us that we're accessing new functional potential. And from a bioprospecting standpoint, that increases the chance of identifying novel functional strains. And yeah, I just want to mention these are really fun to analyze and see these results so far. So to go back to the original slide on IPM, so we're really aiming to move from discovery to application. And so this includes collaboration with industry partners, such as BugBiome, to develop these practical solutions that are aligned with IPM. So the goal is not necessarily to replace chemistry entirely, but reduce reliance maybe on high-risk inputs through biologically informed management. So moving from discovery to implementation in the UK, I think it's also important to note that it requires coordination and collaboration. And so I do want to highlight that there is a new UK microbiome network. So this is to connect researchers across the UK working on microbial functions, tools, and technologies, as well as applications towards sustainable agriculture. Like I said, collaboration is really important if we're going to get anywhere with this. So if you are interested in joining the network, there's a QR code here on the left. Also new is we are holding a seminar series, of which the first will be held in a few weeks on the 20th of February. So this includes speakers both from academia and industry. So the QR code on the right will bring you to the event page. Wait a second for that, I'll take a drink. And so finally, I just want to thank RSB for having this seminar and asking me to speak, acknowledge the collaborators from the USDA, the Hutton, and the students who contributed to this work. I also want to acknowledge BugBiome because I think it's really important that we have this alignment between research and industry towards developing new IPM strategies. And I'm happy to hear from Alicia next, who will continue this discussion because, like I said, translating discovery into field solutions really depends on these collaborations and partnerships like this. So, thank you.
Thanks, Lorena, for that talk. And yeah, thanks, Alicia, for putting your camera on. So, brief intro to Alicia is that she will outline her journey to founding BugBiome, highlighting key steps such as early internships and the shift from skin microbiome and mosquito research to developing crop protection solutions. She will also discuss the importance of culturing diverse microbes, how these are sourced, why diversity matters, and explain why conventional insecticides are becoming ineffective, underscoring the urgent need for sustainable next generation alternatives. So, Alicia, if you'd like to share your slides.
Great. Hopefully everyone can see this. So hello, I'm Alicia Showering. I am the CEO and co-founder of BugBiome. And what we do is discover insecticides from microbes. So we're trying to allow the insects to show us where the insecticidal activity is, to find no insecticides for all the reasons Lorena's just outlined in terms of current pest control. So my favourite insect fact is that there's 1 billion insects per person on Earth. So that's a huge number of insects that farmers have to deal with every single day in protecting their crops. These insects can result in up to 50% crop yield losses, both through direct feeding, but also through the viruses they can transmit. So the insects are becoming harder to control than they ever have in the past. And that's due to increases in pest resistance as a result of less new chemicals coming to market. And that's because new chemical insecticides cost $300 million and take 12 years to get to market, because there's lots of regulatory challenges to be able to show they're only safe towards people and the planet, as well as things like off-target insects like bees. So the time is coming where we're all starting to realise that we need actives beyond traditional chemistry, which is where biological discovery comes to being an alternative. But the current hit rates on finding new actives from collections of microbes, like Lorena's just described, are still very low. So what I'm going to be talking about today is how at BugBiome we try to increase this hit rate in how we find new actives against the insects and translate those into products that are useful in the field. So at the core of this is our platform that has 3 main steps I'm going to talk in a bit more detail about in a minute. The first step is around the microbial bank construction. So we've heard from Lorena about some of the methods we've been working on to increase the diversity of microbes that we can get into the top of our pipeline. And that's key to us on the front end, is getting these diverse microbes. We're not obsessed with crazy numbers of microbes because that makes it difficult for us with our actual insect screening if we go for too crazy numbers. So we're more interested in diverse microbes that are likely to produce diverse secondary metabolites. And then the key parts are understanding the profile of the microbe. So how can we link the microbe to how it metabolizes different substrates and how it impacts insects by looking at the response of the insect to that microbe. So this allows us to find new actives that others miss, and with these, we're developing products from them. So those are insecticide products that could be used either in combination with current chemistries that we have. So the interest there is to find new biological actives that work in different ways to traditional chemistry to prevent resistance. And we're very interested in partnering with the agchem companies and biologicals players that have the distribution and are selling products to farmers. So we're very much leaning into being good at discovery of new insecticides and what's the most efficient way to bring those to farmers is to work with that current value chain. So first of all, microbial bank construction. So we had a nice introduction of my move from human skin microbiome into plant microbiome. And that was all around an interest in how we can protect from various insects that impact crops. And it seemed to me an obvious thing coming into this space to look at the phylosphere, so the plant leaf microbiome, as somewhere to find new actives. That's where many of the reasons that Lorena mentioned, so things like stress tolerance, like it's a really low nutrient, harsh environment on the leaf surface. So if a microbe can survive there, it's likely to thrive when we apply it as a product. There's 2 other areas that we had as key hypothesis coming into this. One was marine microbes. So this was around marine microbes are not naturally exposed to insects. So potentially there's something interesting there in their secondary metabolites, and they have really interesting ways they've had to develop to survive in that harsh, low nutrient, salty sea environment. And the final one is insects' own microbiome. So in the mosquito world, there's lots of work that's done in terms of mining the microbes that are naturally within and on mosquitoes to find new actives. And we found things like Wolbachia to protect from dengue. So I'm very interested in the insect microbiome, and we've done a little bit of bioprospecting of that. So why is this bank of microbes different? Well, there's lots of people out there bioprospecting from soil. And we were very clear from the start they wanted to look in different areas to find diversity that wasn't usually screened on insects. So all these hypotheses were around evolution has already done the selection for us. So hopefully we are increasing that diversity on the front end that will help with getting new interesting hits that other people haven't found. So not just another bacillus, which is what we hear all the time. And then I thought this was a nice figure in terms of what we're really talking about in this space is the natural chemistry from the microbes as new actives. And there's a huge diversity of natural chemistry to be discovered. And that's a much, much bigger space than the current synthetic chemical libraries that the ag chem companies are focused on. And just looking at things like looking at the genetics that have been analysed of microbes, there's over 500,000 catalogue potential chemistries from microbes that haven't been screened enough. And when we're accessing new microbes that haven't been cultured in the past, for example, adding the insect extracts to that media to help getting the diversity in, we think we're likely to find things that people haven't screened in the past, which is super exciting. So getting into the main bits of how we do our screening, the first step is metabolic fingerprinting. So this tool allows us to reduce the number of microbes down from, you might have about 1,000 down to 100 that you can actually handle those numbers on insect assays, which we're going to get to in a second. And what we've developed is custom substrate panels that select for metabolic traits that are linked to insecticidal activity. And this allows us to work with much bigger microbial banks. So they can be our own internal banks. I think we've got about 4,000 microbes now, but also some of our partner banks have 10,000 plus microbes, which are far too many to screen on manual insect assays. So this helps us to prioritize strains to take forward to insect assays. So it helps with that higher hit rate, lower costs, and to make faster decisions. So not all microbes deserve the same attention was our theories here. So unlike traditional screening where you bash everything onto an insect, we're doing a filter before we get to that step. And a little bit of feasibility data from this. So we've identified that some microbes are generalists, so they use lots of different substrates on this panel, whilst others are specialists. And we can cluster strains based on their metabolic profile. And this isn't just linked to taxonomy. So we've got two key clusters on this figure. The orange cluster contains strains with little or no insecticidal activity. And then the green cluster includes all of our hits that we've got in greenhouse trials for aphids. So we've seen nice separation between those two groups that helps us to prioritize micros to the next step. And that is the behavioral screening. So I think a lot of my team has spent various time doing manual insect assays at some point in their past careers. And in the big ag chem companies, the state of the art is sieving aphids into plates of chemistry, leaving them for a few days and saying, are the insects live or dead at the end of this? And with microbes, you tend to see a bit more of a subtle signal. So that traditional screening doesn't make sense because you're going to miss actives that do something, and that could be something useful. For example, reducing fecundity, so reproduction to reduce the population. It could be repellent behavior or anti-feedant behavior. So you're stopping the insect from feeding on the crop. And that can ultimately result in the insect dying, but it can also prevent the transmission of the virus, which in a lot of these contexts, aphids and sugar beet, for example, we're just trying to prevent the virus yellow transmission. So this video capture allows us to look at multiple behaviors and compare to normal insect behavior to see what changes there are, which helps us to find different modes of action that are not that traditional mortality only modes of action that others will miss in their chemistry screening. So I've just got a bit of example data here of how we visualize the videos that we generate. So we can create fingerprints of this behavior. So each of these figures has 7 different behaviors we've been tracking in insect larvae. And we've got on the left a negative control, in the middle a microbe, and on the right a chemistry. And what we can see in the microbe in the middle is we've got a much higher proportion of time spent doing the bend behavior. So this allows us to look at these and identify, based on those fingerprints, what are the differences between the negative controls versus the things that we're screening. And the hope here is by running all the known modes of action, so as IRAC classifications of insecticides, can we start to classify fingerprints based on how similar they are to known mechanisms of action, and ultimately help with resistance management by finding actives with entirely new modes of action. That's the future for BugBiome. But cracking the microbial black box was what we came into this space. So phenotyping, actually allowing the insect to show where the activity is, can outperform sequencing, because when you're looking at sequencing, you're going to find mechanisms of action that are already known, whereas we're interested in finding new mechanisms. So here is a figure that shows, by looking at that lethal mortality behaviour, you find the actives in pink. But by looking beyond that to look at behaviour, you can identify all these dark green behavioural shifts that can potentially be new actives that when you bring them through into more screening, you can find something that is helpful to protect the crop, but would have been missed in traditional screening. So I was quickly going to mention my RSB student from last summer, Leah, who did an interesting project bringing in fungal screening to our usual screening pipeline last summer. What she did was grow these fungi up into broths and apply them to two model insects that we work with, mealworms, so a coleopteran, and wax moths, a lepidopteran, and then assess their mortality over time. And what she identified was 4 fungal strains that had some efficacy against mealworms and 2 against wax moths. What we're doing with these now is doing phenotypic screening to understand how do these fungi impact the insects that she was screening on, and is that efficacy good enough to be able to take those forward towards future product developments. There's various pest targets that we've been working with. So aphids was the insect target we set out to work with initially. We are currently running many greenhouse trials on aphids, with field trials planned for later in the year. But there's lots of insects that farmers tell us all the time that they're interested in. So this is a kind of summary of the ones that we are working with at the moment. So we are starting to look at other aphid species this year. We are currently trapping some pea and bean weevil, cabbage stem flea beetle, and wireworms, all interesting UK relevant pests. We've been doing some screening on diamondback moth for a while, but there's lots of other pests that are within these different orders of insects that we'll be looking to do screening on in the future. And I said I would quickly recap some of how I got to starting BugBiome, as I met very few people running companies before I decided to give it a go back in 2023. So I am from Bristol, if you can't tell from the terrible Somerset accent that I've not been able to shake fully, but hopefully you've been able to understand me. And I went to state school in the UK, so I remember rocking up to sixth form one day and somebody saying, oh yeah, you should go to a Russell Group university, and that sounded like a good idea to me. So I had a look at the universities on that list. I drew a 3-hour ring around Bristol to make sure that my parents couldn't drop in to see me, and Liverpool seemed like a good idea. And it definitely was I had a great time at Liverpool, very glad I went there. And the course that I chose was biological science. I didn't realise when I went to do it that this course allowed you to try things, and if you didn't like it, you didn't have to do it again. But if you did like it, like for me, microbiology, you could then do that every year of the course. So this really gave me a good grounding, and I could try different things, see what I was interested in, and continue it if I wanted to. So at the end of that, I realised I was definitely interested in microbiology, insect biology, and public health. One thing to note from that time was I did an internship between the second and third years of my undergrad, which was funded by Microbiology Society International, with an awesome professor, Alan McCarthy, who agreed to have me in his lab for 8 weeks. And that was really super helpful to be able to work on a project that I was interested in, actually get some lab experience. At the end of that, I thought, oh, let's do a PhD. This seems like interesting, trying various things in the lab. So I saw a project advertised that was looking at the human skin microbiome and attractiveness to mosquitoes. I thought that seemed like a cool project. So threw an application in. Was not expecting to get an interview, as I didn't have a master's at that point, but was fortunate to get one and was offered a 1+3, which was a master's combined PhD. And I suggested maybe I could do entomology as that master's. I thought that'd be a pretty fun year. They suggested medical statistics, which I wasn't too happy about, So we compromised on epidemiology, and on reflection, that was a super useful year to understand things around statistics that became very useful during my PhD, doing some of that molecular analysis of sequencing data. I took part in lots of extras. Anything that got emailed to me, I'd usually say yes to. That included industry placements, teaching, taking part in young entrepreneur scheme, pre-accelerator programs. And through that, I kept coming back to the same idea of surely we can use microbes as alternatives to chemistry as an active. I was thinking about mosquito repellents for a long time, and got some feedback through those different accelerators around there's actually lots of insects beyond mosquitoes, and whether or not there would be potential to use the same ideas to be able to find new solutions against them. So when I rounded up my PhD in 2023, I was fortunate to be awarded some Innovate UK funding. I raised our first round of investment from angel investors, and we got a place at the BugBiome campus on the Accelerate programme, which gave us a free lab to get us started that had all the equipment we needed. And Cambridge is a great place to get started, but John Innes was somewhere that has always been spoken about to us in terms of doing microbes, plants and insects. So we span into the John Innes Centre last year. There's lots more I could say about pre-accelerators, programmes, funding, etc. But I thought it'd be helpful just to put a few of the logos on the slide of people that have supported us over the last 2 years. And if anyone's got any questions around starting a company or is interested in collaborations in insecticides, we're always interested in finding collaborations with people who have interesting strain banks that might make sense for us to screen, or supporting student placements. Thank you for listening.
Excellent. Thank you both very much. Excellent series of linked talks with absolute fundamental science, but then leading forward to the requirement to translate that into real impact. So loads of questions that I've got, but we've got a couple already in the Q&A, and I asked everybody, chuck your questions in there, because there's no such thing as a stupid question, and this is more of a discussion than an interrogation. Don't worry, Lorena, it's all right. So I'll start with the question that came in first, which is quite a obviously topical one. So how do you think climate change will impact on the results? And I think they're specifically mentioned enrichment. And does this affect your experimental design or your analysis?
I can speak to that one. So yeah, if we talk about enrichment specifically, there have been other instances where we will design based on like the influence of temperature. So if we think, this is topical for climate change, just because if there are increasing temperatures in the UK, for example, how is this going to change the microbiome? It definitely will. And then we've shown that there are changes in taxa that are present at various increments of temperature change. So you can enrich in cold temperatures, and you can enrich for certain microbes in warmer ones. And this is an approach that we have taken to expand diversity in our culture collection.
Okay, anything to add to that, Alicia?
I think just linked to that in terms of that enrichment, it's also interesting if you compare the UK climate, for example, to places where biologicals are being much more successful. Brazil, for example, is a place where all farmers are using biologicals because chemistry's failed. It doesn't work anymore on the pests there. So actually there's a lot of things we think about of enrichment to make biologicals that are more suitable to the UK climate, where it's actually more difficult because it's colder, it's rainy, it's not nice throughout the winter, especially when you're thinking about live microbes.
Great, thanks. So I've got a comment here from a PhD student in Burkina Faso in Africa, Namwin Yele. And she's saying, she'd obviously like to collaborate, but makes the point that in that part of the world, there's lots of limitations technologically. I mean, it's a comment rather than a question, but obviously we've got all these fantastic technologies, but in other parts of the world where perhaps these are needed more, we haven't got those. So just a comment there from a PhD student. Yep.
Yeah, and in my PhD, I was at the London School of Hygiene and Tropical Medicine, so we had research sites out in Gambia, and that was, yeah, we had better equipment there than what we had in London. So I intended to do all of my molecular analysis there, but COVID put a stop to that. But I think there definitely are potentials for collaboration out there, and at institutes like LSHDM, it's very much on the agenda to be collaborating in country. So I think anything to do with public health, there should be opportunities to collaborate there. I might just put you in contact with some people if you share a bit more in the chat about specifically the area you're working in.
Great. Joanne asks, there's a popular hypothesis for the formation of disease-suppressive plant microbiomes is the cry for help model, where diseased plants recruit or select rhizosphere microbes by producing secondary metabolites. Have you considered exploiting this concept by adding these metabolites to your communities and see whether that improves the outcome? Is that Lorena?
Yeah, I can comment on that. So yes, we also so we do these antagonism assays like I showed you on the plate, but we also do volatile organic compound assays. So we look to see if any of these metabolites from these microbes can antagonize. And actually some of that work back in Fargo was looking at this specifically and trying to figure out what these organic compounds are and if they're being produced more so in the presence of the pathogen. And so, yes, this is something that we definitely do explore. Yeah.
Okay, cool. There's a question here from Elizabeth about, which I'll add a rider to, how are the microbial samples collected from the insects of interest? And my interest, where I used to work on aphids, is how does the different feeding guilds of insect affect the microbiomes?
Yeah,
that's really
so
Alicia.
yeah, so the old question, Jeremy, I never thought too much about that one, to be honest. But getting the microbes from the insects, a lot of it has been the usual kind of culturing methods, so different medias, temperatures, etc., that we've talked about today. And then either taking the microbiome off the outside of the insect or sterilizing the outside and then crushing the insect up as you would with a plant sample or anything else. And we have done, I think sometimes you see insects in the wild, for example, that have died and you can see some fungi on them or something like that. So that was also something we explored, was like getting soil samples and getting mealworms from there that you could quite visibly see have been impacted by microbes and culturing them that way. So we tried various things, but we're very much interested in other methods for that. It's not an area we've done much development on. So yeah, definitely open to different ideas. And I think the same with kind of crop pest targets. We definitely thought a lot about that in terms of target in the crop we're interested in. So that might be aphids and sugar beet, actually target in the sugar beet microbiome. And I think the same with the aphids, but we've not really ever done much bioprospecting around finding lots of natural aphids in the field and seeing what interesting things we can get from there. So that's definitely an opportunity, I think.
Yeah, I mean, my thought was, EPG or something, because the complexity of phloem location in aphids and the different points. I think you alluded to it to a certain extent with your little snail diagrams. I thought that's really, really cute. Okay, Josephine asks, if you observe reduced microbiome diversity associated with disease occurrence, have you considered increasing that diversity as an alternative to looking for new chemical, in inverted commas, modes of action? I don't know who wants to pick that one up.
I think I was going to say, like we honestly, we've had lots of arguments to me and my co-founder around live microbes versus lysates or metabolites from microbes. And I think even just thinking about that, live microbes and actually getting them to establish is hard, let alone consortia live microbes. And then the cost of one microbe is expensive, let alone a mixture. So just thinking from the operational point of that, I think that's much tougher to do, and it's definitely put me off it. So we're very much focused on single microbes, extracts from that, than we are trying to do that.
Cool. And sorry, Lorena, do you want to come in on that?
Well, I was just going to comment that I think it's important to a lot of what the regulatory component of that. So how consistent your product is over time. So I think looking for the metabolites and products might be easier ultimately for a product than using whole microbes themselves. But
I'll go to a question now from Thelema, which seems to pick up on that and touches on that regulation issue. So, are there any potential risks associated with these, and how do they compare with the chemical alternatives? So, such as risks to farm workers, and so on. Presumably, that ties to getting these things approved.
Yeah. So it is. There are accelerated pathways for biological products, even in the EU, but they're not super fast. So it's still tough. It's still very expensive. There's still a lot of ecotox safety you need to prove. And microbes are more specific than chemistry is, or tend to be more specific. So in biological insecticides tend to work on a more specific insect target than things like neonicotinoids that really worked on a wide spectrum of things, and that's when you start to have those off-target impacts. So inherently should be safer based on the evidence out there, but there's still always risks. You can always find the toxic microbes. So for us, we have an early bioinformatics screening pipeline that we've implemented internally. And if we get a red flag on that, then we'll drop the microbe instead of continuing with it. So I think that there's always pros and cons to these things, and I can assure you the regulation's still tough in the insecticide space. That's not so true in biostimulants. But if you're saying that you're having an impact on an insect, then there's definitely it's hard, expensive.
I think it's important also to say that currently in the UK, I've had to read quite a bit lately on regulation here. And biologicals, if it is a biological control agent, it is regulated currently the same way as fungicides. So it has to go through the same health and safety executive paperwork and all of that. But there is a lot of talk of that changing, and like Alicia mentioned, kind of getting streamlined and maybe being looked at differently. So hopefully that will happen.
Yeah. Okay, Helen asks, and it's really interesting questions. Have you done any work with companion plants having their own phylosphere that can attract insects, like say molasses grasses and so on, stem borers?
So I wouldn't say I've worked directly with companion plants, but one thing that we are looking at are wild relatives of crops and their phylosphere. So I've looked at, within the UK, wild barley phylosphere, and looking not only phylosphere, but endosphere as well. So looking for microbes that are intimately associated with those and seeing if we can establish them in domesticated barley, for example.
Yeah, and for us, we've not looked at that. We've done work on crop versus non-crop, and that's about as exciting as it's got. But that's definitely an interesting idea.
Nice, thanks. And then Jane asks a simple question, which is close to my heart, is from Bifor. Any current research into forestry-related pests?
We're not doing any work on forestry related pests at the moment. Very much, horticulture, row crops is where our focus has been. But there was a Scottish startup that was funded, had a good fundraise a couple of weeks back that was doing forestry microbiome. So maybe they'll head out into that space.
I wonder if Jane, if you chat in the chat, what particular trees and disease were you thinking of? And we'll just pick that up. Okay, anonymous attendee, a question for Alicia. How do you address the challenges of upscaling interesting gene clusters that may be silent within microbes or require complex conditions to be produced?
Yeah, so we're not genomics first. So if we do find an interesting gene cluster, it'd be more likely to be to do with the second one. So having done things to the media to be able to get it to be produced for us to find it. There obviously are ways that you can use CRISPR and upregulate gene clusters, but that's not in our remit for what we do. But companies like BugBiome, where I used to work, were definitely more focused on that. kind of natural product engineering side of finding things that are there in the genome, and there's lots of people doing that. For us, it's very much more focused on the phenotypic side of things and finding actives up.
Cool. Okay, how do you address mutations of the screen microbiomes and potential for unintended consequences? Ooh, nobody wants to pick that one up. I think anonymous attendees got you there.
Well, do we mean intentional mutations? I guess I'm not quite understanding.
I think the unintended means the
they said
yeah,
unintended mutations.
Yeah.
So sort of focus, maybe forcing mutations based on putting it in a new environment. Is that kind of the thought?
Maybe we can ask them to clarify that, and if it comes back, I'll come back to it. Oscar asked a question, an interesting one as well. Considered using biological vectors to enhance inoculation, such as earthworms. Darwin would have been very pleased. And that also might be interesting. Is it currently just spraying that you use?
Yeah, no, we've not thought about that. So interesting idea. Yeah, and we are doing foliar applications. That's our main focus. Yeah, definitely. I'm excited by seed treatments, but I think that comes later down the line.
And how recalcitrant or positive are farmers about this? I mean, are they keen to use these biologicals? Are they resistant to change?
I honestly think there's big differences in farmer attitudes between different countries. So at the moment, I'm doing my basis agronomy training. So it's interesting to be in a room of agronomists and farmers and get that direct feedback. And I think we're more kind of reliant on chemistry here than we are in countries like Brazil, where there's been that swap to biologicals. And I think there's more work to be done here. I don't know if some of that is what we touched on earlier, to do with the fact that biologicals, especially live microbes, tend to do better in those hotter environments. But I think, we still have work to do in this country. And I think that the regulation makes things more difficult because we have fewer biologicals, especially fungicides and insecticides, that are on the market, and the things that are available are biostimulants, which gets this reputation of snake oil. So I think that getting farmers on side, and I think things like the adopt grants that are coming through are good opportunities to try to do that. And for us, while we're working with farmers and showing the results, then they're interested. I understand the skepticism, and also our kind of reliance on chemistry right now. Just when you think about the cost of goods and get to that point where we have better products like this, is still it's not tomorrow.
Yeah, I mean, it's sort of going back to a previous sort of part of the discussion. I mean, is it possible that the regulatory framework might persuade them more, or is it always going to be tight for both ways? I think Lorena touched on that earlier.
I think, well, as things are withdrawn, but also as resistance forms. So, for example, black leg of potato, there aren't any chemicals that we can use on it, and so when you have that problem, I think you'll work with anything that is suggested to you. So I think as disease pressures increase, maybe with climate change, and then regulation gets tighter, or maybe there's less chemistries to use, that it'll have to be adopted more.
Yeah. And a technical point, and I may have missed this in some of the talk. So you're talking a lot about phenotyping. How close are you to getting to really high throughput phenotyping, or are we close to that, or are the things that are coming along that might help?
Yeah, for us, we haven't focused on that like throughput of people like Syngenta. Our goal is not to screen like 20,000 microbes a week. You need very different tech to be able to do that, and that probably could be possible. But for us, it's been more around having clearer reasoning for things, and I think we do a lot around kind of target product profiles and trying to think about products. So alongside that, actives, back to this same point of like products have to work for farmers, so they need to work with the tech that we actually have on farm. And when you start to look at dose rates, etc., there's lots of work we need to do around those products to make sure they fit into the systems that we have, to make that then it's not a behavioral change, it's just a different product, and you're using product X instead of the usual chemistry.
Okay, great. I think I've heard the clock tower on campus just strike the hour, which tells me that our time is done. I'll just tell Lorena, at one point there were over 130 people. Lorena didn't want to know at the beginning how many people were listening, but I think you did a marvelous job, and thank you as well, Alicia. Really interesting discussion. I've got loads more questions I could ask, but unfortunately, my clock tower has told me it's time to close the meeting. So thank you both very much. And I hope everyone enjoyed that, and you'll follow up with questions and so on. Beth has just put a message there. There's a short survey there. If people could fill that in, it helps us target these talks and think about what's good for people and what's good for the whole plant health community, which is what this is all about. So once again, thank you to our speakers, and thank you to you all for your attention and your questions. Makes it easy to be chair. Thank you all, and see you all again soon.